Okay guys, let's go ahead and get started. So, today is the review session. But before we get started with that, let's just briefly talk about the exam and how it's structured. Okay? There will be 3 lecture exams and they all will be structured in the same way. Okay? The only thing that changes is the material that's covered in the exam. So, there will be 20 matching questions. And these are worth 1 point each. 10 multiple choice. Again, this will be 1 point each. 5 short answer. That gets worse. 2 points each. And then 2 essay.
What?
5 points each. You can imagine, you'd be surprised. And there will be extra credit of 1 to 2 points, depending on how generous I feel. So I've heard everyone so far, or pretty much everyone in the room, complain about short answer essay. You will learn that these will be your friend. Okay? I am relatively lenient when I grade. However, I can't be lenient with matching your multiple choice. It's either right or it's wrong. Okay? With these guys, as long as you're kind of in the right track, you're good to go. Okay? Also keep in mind this isn't an English class, this is science. So it's short answer just because that's what the type of question is, but it doesn't have to be complete sentences, it can be bullet lists, it can be cartoons, whatever. As long as it makes sense and you answer the question. Okay? For these 2 up here, this will be with Scantron. Okay? So make sure you bring with you a pencil. Okay? In theory, black pen should work, but just bring pencil to be on the safe side, okay? Now for matching, because there's 20 different choices, you have to get a little creative. So some of the answers may have a series of letters next to them. Let's say A, B, C. That is what you would fill in for that answer on this Scantron, okay? So if you think the answer is A, B, C for that number, you would then fill A, B, and C. In the same line. Okay? Any questions about the exam? Structure? Okay, good.
Do we still have lab before the exam?
Yes.
Exam?
Sure. Got no problem with that.
Oh, sorry, what did she ask?
Take the exam before. Everyone want to do that? Or do you want to do it after that?
Yeah.
Okay, raise your hand before.
The only thing is that you got 2 people that might be late because they get work.
Yeah, I can see that.
That's
Unless
all
it's
you-
unanimous, we got to stick with the rules.
Sorry.
So we're going to do lecture or lab and then lecture. Sorry guys.
Sorry about that.
All right. On with the study section, okay? So the way that I structure this is I go through each chapter and I pick out the important bits, okay? I do ask a lot of questions because I want to get you guys involved. So please don't hesitate just to shout out the answer, okay? The longer it takes you guys to respond, the longer we are here, okay? Because I will wait until someone says something. Don't be afraid to say something wrong. I'm not going to shoot you down. I will just tell you why you're wrong, okay? So, that said, let's get going, okay? So chapter 1, as I said when I first gave this, most of this is just for your information, right? To give you an idea about what microbiology is. But there are some key important parts that we talked about that I think will be useful on the exam. So first off, you should know the basic groups of microorganisms, okay? So bacteria, algae, protozoan, fungi, viruses, and helminths, okay? And you should have an idea of what those are, right? So for example, a protozoan is a single-celled animal. A helminth is a multicellular animal that looks like a worm, okay? And again, we've talked about most of these in more detail, so that shouldn't be too hard for you to understand. This slide is a nice summary slide of why microorganisms are important for us. Okay? So have a good idea about why we utilize microorganisms, but also how microorganisms cause disease, okay? And why this is important for us to understand. So we care about microbes because if they cause disease, they may kill us, and we could potentially stop that if we understand about that. Slide 22, this is a great summary of the differences between prokaryotes and eukaryotes. Okay? We've talked about this not only in this chapter, but in chapter 4 and chapter 5. So I can guarantee you there will be a question on the exam about the differences between prokaryotes and eukaryotes. Can anyone give me an example of a difference between a prokaryote and a eukaryote?
No organelles in the
So no organelle in prokaryotes. Anything else?
No nucleus.
No nucleus? Anything else?
Eukaryotes are sometimes bigger.
Eukaryotes are bigger, right? Anything else?
Eukaryotes don't have a cell wall.
Well, some eukaryotes do. So plants and fungi can have cell walls. People get tripped up on that all the time, though. Anything else? There's literally tens of these.
All prokaryotes are microorganisms.
All prokaryotes are microorganisms. Good. Anything else? Chromosome. Okay, so prokaryotes have a single circular chromosome where eukaryotes have multiple chromosomes that are linear. Okay. So there are 5 examples. So make sure you have a handful of those in your memory. Okay. You can regurgitate them. Now, when we did talk about chapter 1, I picked out a slide, this one in particular, and said this would be something that may be on the exam. Okay. And this talks about 2 important people in microbiology, Louis Pasteur and Robert Koch. So why is Louis Pasteur important for microbiology?
He was the father of microbiology.
He was the father of microbiology, and why did we say that? He came up with the germ theory, right? That basically states when you get sick, it's not magic, it's just you came into contact with a small organism you cannot see. Okay? It inhabited you and made you ill. What about Robert Koch? Why do we care about Robert Koch?
He verified the germ theory.
He verified the germ theory in human disease. Okay? And he did that using Koch's postulates. Does anyone remember what Koch's postulates are? Or how would I go about determining if a pathogen causes disease in humans? Yep, so the first thing you do is isolate the causative organism, right? So you take it, you streak it on a plate, and you grow individual colonies. You then take the causative organism, and what do you do with it? You infect someone or something else, right? And then you wait to see if they get the same signs and symptoms. And that means you're on the right track. But you're not done. You then have to re-isolate it from that second individual, and then compare and contrast it to the primary or first organism. It looks the same, acts the same, and causes the same disease. Congratulations, that's the organism that caused that disease. Okay? Can you guys think of any problems with Koch's postulates?
It's ethically wrong.
So ethically, it could be wrong, right? If you have to infect a healthy human, that's usually a bad thing, right? Anything else?
Can't grow in the lab and you can't really do it.
Right, so it has to be able to grow in the lab. You have to be able to culture it, because that's how you isolate. If you can't grow it in the lab, it will not work. Okay, so there's some problems with this, but we do have ways around it. Okay, good. So that's chapter 1. Chapter 4. So we're skipping chapter 3 because that will be in the lab exam. Okay, remember those are mostly laboratory techniques. So, for chapter 4, we deal with prokaryotes. So here's just an example of how prokaryotes and eukaryotes are different. And in this example, we're showing how much smaller prokaryotes are compared to eukaryotes. Does anyone remember why this is true? Why do prokaryotes need to be smaller? They don't have organelles, so they don't have the ability of moving things around big spaces. So they have to have a small space because they just can't maintain it. Good. All right, so this is a nice summary slide that lists all of the different bits and pieces within a prokaryote. Okay? I like this slide because it gives you a lot of white space where you can take notes, and you can also test yourself to see how much of this you remember. Okay? What I expect you to know for the exam is a one-sentence definition of each one of these that describes what it does and where it is. Okay? So, let's start off with flagella. What is a flagella?
Tail.
It's a tail that propels the organism, right? And how does it move in prokaryotes? Spins like a motor, right? So it spins. Good. Pili, what is pili?
It's used for replication, or like,
It's used for
yeah.
Attachment and genetic
transfer of genetic material. Right, so you're kind of on the right track. One easy way to remember this is P. pili, P. penis. It's basically a bacterial penis, okay? It injects itself into a cell and then transfers genetic material. Fimbriae, what do fimbriae do?
They attach
They attach, right? They're little hair-like structures that attach and stick the organism to a place. Good. Glycocalyx, what's the glycocalyx?
It's the outside
It's the capsule or slime layer. Slimy layer on the outside, right? And its job is to protect the organism. And what is it really good at protecting against? Phagocytosis, right, or gobbling up of the organism by our immune cells. Okay, so by blocking this, they can survive in our body longer. All right, onto the cell envelope. Next, we have outer membrane. What organisms do we find the outer membrane on? It's in parentheses because it's not on everyone. So we've got gram-positive and gram-negative. Who's got 2 layers?
Positive.
Who's got 3 layers? So who do you think has the outer membrane, which is that third additional layer? Gram-negatives, okay? So gram-negatives have the outer membrane. Does anyone remember what the outer membrane's made of? It's a phospholipid layer similar to the cell membrane, okay? So it looks a lot like the cell membrane, but on the outside of the cell. What about the cell wall? What does the cell wall do for the organism? Gives it structure, rigidity. What is it made out of? Peptidoglycan. Is that the only thing it can be made out of? What's the other thing?
Proteins? Just kidding.
Begins with an M. Begins in Mycolic acid. Mycolic acid. Okay. So it can be peptidoglycan or mycolic acid. All right. And if you are a gram-positive organism, how thick is your cell wall?
Very thick.
Thick. Right. And if you're a gram-negative organism, how thick is your cell wall? Thin. Good. Cell membrane. What's the cell membrane made out of? Phospholipids. Good. And what does it do for the organism?
So it's like a barrier, so like, all the proteins in and out.
Right. So it's a barrier from the environment. It determines what goes in and out of the cell. Okay. This is what actually creates the cell. Good. Internal. Cytoplasm. What does the cytoplasm do?
It's the fluid that
Fluid, everything floats in. Otherwise, it does nothing. Okay. Ribosomes. What are ribosomes?
make proteins.
They make proteins, right? So they're structures that allow for translation. Inclusions. What are inclusions?
Compacted masses of viruses or cell organisms.
Could be,
just
but
say-
they're found in healthy cells as well.
They store.
Do they allow for diffusion of fluids and things like that you were saying?
No, not so much.
Okay.
They store like vacuoles.
Store crack, right? They're storage places. So they're basically holes in the cytoplasm where crack gets stored.
Now you can give Anthony the right store stuff, crap on the tusk.
Yeah? Give them right shit, I don't care. Inclusions, all right, we got that one. Nucleoid of the chromosome. What is the chromosome? Genetic material, DNA, right? And what does the chromosome look like in prokaryotes?
Like a ball.
Circular, and it's usually singular, right? So by itself. Good. Actin cytoskeleton, what is this? It's actually in the name.
provides the support for the cell
It-
structure.
The other portion.
Skeleton.
Cytoskeleton. Cyto means cell, skeleton is internal structure, okay? Just like our skeleton. So it provides internal structure to the cells. And then endospore,
So now where things get through.
They go in when they're under threat, they kind of go.
right, so when conditions are bad, they kind of wall themselves off and stop being metabolically active, okay? And this is where they sit until conditions get better. Good, okay? So this is basically all the major bits and pieces within a prokaryote. Now, I'm going to skip over a lot of slides, and that's because we've already talked about most of these things. That does not mean they won't be on the exam. If it's on this piece of paper or this slide, know that, okay? I skip over them now, it just means we've already talked about them, there's nothing extra I want to add. This is another good slide if you are more of a picture person. You can actually see where things are. And this also has the nice one-sentence definition to go along with each structure.
Are you gonna put an explanation to the picture, or are you just gonna know?
So there will be no pictures, right? You don't have to do that. But that's, everyone's different in the way that they learn, right? Like, I could tell you something and you can learn that way, or you have to see it and visualize it. It just depends on the individual. Okay, so flagellum. One thing that we didn't really talk about with this is how they are arranged on the cell, okay? You will need to know this for this exam as well as potentially the practical. So if I say monotrichous, what does monotrichous mean?
One.
Single flagellum, right? What about lephiditrichous? Multiple coming from one place, okay, or one pole. What about amphitrichous?
One on each end.
One on each end, or sometimes multiples on each end, but the key here is they're on either end. And peritrichous?
All over.
All over.
All over. Does anyone remember what's the most common? Right.
Paired.
Peritrichous, right. So this guy is by far the most common. What is chemotaxis? We talked a little bit about this in class.
That's like when they move because there's a chemical.
Right, they're moving towards a chemical gradient or a chemical signal in hopes that they find where that signal came from and then they get all of it and can utilize it for whatever, okay? And this is important when we talk about flagella because this is how they move towards those chemicals. Good. Now with the glycocalyx, remember it can be organized in one of two ways, slime layer and a capsule. Slime layer is loose, capsule is tight. Okay, it's the big difference between the two. Otherwise, they're the same stuff, polysaccharides. And remember that glycocalyx is important in preventing phagocytosis. It also allows the organisms to stick together, forming a biofilm. All right, so the cell envelope, we've already talked a little bit about this, but I just want to make sure we're all on the same page, okay? Gram-positive organisms have 2 layers, gram-negative organisms have 3 layers, okay? What are the 2 layers in a gram-positive organism? Cell wall and cytoplasmic membrane, and what is the cell wall made out of? Peptidoglycan, and how thick is it? Thick, good. What about gram-negatives? What are the 3 layers in the gram-negative envelope? Outer membrane, cell wall, and cytoplasmic membrane. Good. And how thick is the cell wall? Thin, and it is made out of? Cell wall is made out of? Peptidoglycan. Good. Mycolic acid is going to be found only in Mycobacterium, and they aren't gram-positive or gram-negative. Okay, they're their own little thing. Okay, they're called acid fast. So if it's gram-positive or negative, it's always peptidoglycan, and this is 95% of bacteria. Okay. Now, the outer membrane also has an additional molecule. This molecule is called lipopolysaccharide, also known as LPS. Does anyone remember why we care about LPS in this class?
Acts as an endotoxin.
It can act as an endotoxin, right. And endotoxins are toxins that are released only when a cell dies. So when the gram-negative dies, releases this, gets into our body, and this causes fever. Okay? So LPS is responsible for a lot of fevers. Gram staining, all you need to know for this exam is gram-positive is purple, gram-negative is pink. The actual gram staining procedure and protocol, that's something I would test you in the practical. Okay? Not so much in this exam. And as we said, cell walls can be weird. If they lack a cell wall altogether, they're known as mycoplasmas. And if they use mycolic acid instead of peptidoglycan, they are known as mycobacterium. Okay? So these are what we call non-typical cell walls, and again, it's just a small percentage of prokaryotes. Talk about cell membrane, about cytoplasm, DNA. Oh, we skipped over plasmids. Does anyone remember what a plasmid is?
Circles of DNA.
A circular piece of DNA that's small, and what is it used for?
Doesn't it go into like a daughter cell?
Yep, it's how, this is what gets transferred through the pili. Okay? So it goes from one cell to the next. It's used for transfer of genetic material. Talked about endospore. Okay, and then lastly, slide 53. This has the shapes. That's all I expect you to know for this exam. Okay? We talked about arrangements and other things. Again, that's something I will test you on the practical. Okay? So for this exam, just know the different shapes. Cockeye, bacilli, and spiruli. Okay, good. Chapter 5. Eukaryotes. So again, another slide explaining the differences. Okay, trying to beat this in deep. Alright, so this is a nice summary slide of all the different structures within a eukaryote. Again, there's a lot of white space on this, so you can test yourself and see how much you remember about these. Okay? So let's go through them and see how much you guys know. Starting in the middle, what is this big structure right here? Nucleus. And what does the nucleus do? Protects DNA, right? So it houses the DNA. In the center, we've got this little purple ball of yarn. What does that got? The nucleolus. And what does the nucleolus do?
Produces parts for ribosome.
Right, it makes our RNA or the ribosomes, okay, eventually. And remember, the nucleus is covered with, what's it called? No? The nuclear envelope, okay? And that is basically a phospholipid bilayer, okay, times 2. Outside of the nucleus, you've got all these squiggly lines with dots. What is that? Heard a bunch of stuff. Yeah. Rough endoplasmic reticulum. Okay, and what does it do for the cell? Protein synthesis, right? And that's because it's got all these little dots on there. What are those dots? Ribosomes. And again, what do ribosomes do? Make protein. Good. Next to that, you got these little smokestacks. What are these guys? The smoothie or smooth endoplasmic reticulum. This does 2 things for the cell. What's one of those? Lipid metabolism, or processing fats and lipids. And the other thing?
Synthesizes hormones or
Cleans out.
Yeah, detox, right? It gets rid of any toxic substances that may be in the cell. Down below that, we have this guy. Who's he? Mitochondria. What does the mitochondria do? Powerhouse, right? Chapter 8, cellular respiration. All happens within the mitochondria. Next to him, we got the chloroplast. What does that do? Who do we find the chloroplast in? What kind of animals? Or, excuse me, not animals, by the way, organisms. Plants, right. So what do you think it does? Yep, photosynthesis. This is where CO2 and water and sunlight eventually make glucose. So again, this would not be found in pathogens. And then above that, we've got this guy. Who's he?
Golgi.
Golgi apparatus. And what does the Golgi do?
Pathogens of sorts.
It traffics, right? Moves things around. Okay? And again, the Golgi is one of the reasons why eukaryotes can be so large, because they have the ability of moving stuff around. Alright, so now we'll go into the envelope for eukaryotes. There's either one or two layers. Okay? If it's an animal cell, there's just one layer, and that is the cytoplasmic membrane. If it's a plant or a fungus, there are two layers, the cytoplasmic membrane and the cell wall. Bonus points. Does anyone remember what the cell wall of fungi is made out of? Chitin, right. And what about plants? Cellulose. Okay, so fungal organisms use chitin, and plants use cellulose. All right, external structures. What's this guy? Flagellum, right? And how does it move? Wags like a tail. Okay, so eukaryotes wag, prokaryotes spin. Good. Also cilia, which you couldn't see in the picture. Anyone remember what cilia are? Little hairs that move, and they can move in unison and propel the organism around. Okay? Good. Also glycocalyx, which can be found on eukaryotes as well as prokaryotes. It is virtually identical though. Okay? It's made out of polysaccharides. It can be a slime layer or a capsule, and it serves the same purpose: to protect the organism primarily from phagocytosis or to produce biomass. Talked about this. Vacuoles. Anyone remember what a vacuole is?
Open space.
Open space, right? It's kind of like an inclusion. In this case, it's in eukaryotes. And in the vacuole, you can store prep, right? Liquids, food, whatever. Okay, so those are all the bits and pieces of eukaryotes. Then we went and talked about all these different kingdoms found in eukaryotic, okay? And I talked about a lot of different examples of diseases and organisms and things like that. For this exam, I don't expect you to know that, okay? What I expect you to take from this portion of the talk is essentially some bullet points about each of the kingdoms. Okay, what kind of organisms they are, what do they look like, you know, where are they found, that kind of thing. All the specifics I gave you about certain diseases and things were more just for your own information at that time. Don't worry though, they will come back to haunt you, and for chapter 3 you will need to know what they are. Okay? But for right now, you don't. So, kingdom of fungi. Kingdom of fungi, these organisms are terrestrial, non-photosynthetic, and saprophytic. What does it mean to be saprophytic?
They feed off the dead.
Yep, you eat dead things, right? So these guys are mostly organisms that eat dead stuff. However, occasionally they can cause disease and they become parasites, okay? But it's few and far between. Remember, if they are single-celled, we call them yeast. If they are multicelled, we call them molds. And when they're multicellular molds, they are made up of hyphae, which are strings of cells. And when you have a lot of hyphae connected together, this is what we call the mycelium. Remember, two unique molecules of fungal organisms are eugesterol and chitin. We've already talked about chitin. This is found in the cell wall. And eugesterol is found in the membrane. Okay? Remember, eukaryotes need sterols to keep their membrane together. And for fungi, this is eugesterol.
What does that do again?
The sterol, eugesterol, it keeps the membrane together. It gives it more structure. So, as far as hyphae are concerned, there's two flavors, vegetative and reproductive. Vegetative is used for what?
Feed it.
Feed it, right? These are the guys that will dig down into the nutrient material, digest it, and suck it up into the organism. What about the reproductive hyphae? What do these do? They release spores, right? And spores are just baby fungi, okay, little baby guys. So this is how they reproduce. Remember, yeast are single-celled fungal organisms, okay, and they can be both useful and harmful, okay? It just depends on the species. Again, you don't need to know any specifics. And as far as reproduction is concerned, they can reproduce both asexually and sexually, okay? Unlike prokaryotes. Why do we care about fungi? So fungi are useful for humans, right? They get rid of dead stuff. They also help us make things like beer, wine, alcohol in general, cheese, a whole bunch of stuff. But they can occasionally cause disease. But remember that when they cause disease in humans, it's usually because your immune system is weakened. It's not something that normally happens, at least in a severe type of disease. All right, so now on to the protists. Protists, there's two subkingdoms. Algae, which are single-celled plants, and protozoa, which are single-celled animal-like cells. Okay? For algae, all you need to know is that they are single-celled plants and they are not pathogenic. For protozoans, okay, we have single-celled animal-like organisms, most of which are free-living, which means they do not harm humans. Okay? However, there are a very small percentage of protozoans that can cause disease and act as parasites. Okay? And remember, their life cycle has both a trophozoite and a cyst stage. Trophozoites, metabolically active. Cyst is what they do when conditions get bad. They wall themselves off in like a little cocoon. And lastly, we classify these guys based on how they move. Pseudopods, cilia, flagella, or no movement at all. Okay? Out of those four groups, which one's the most important for us as far as human pathogens are concerned? No movement, cilia, flagella, or pseudopods?
Flagella.
No movement. Why? Because they are all parasites. Because they lack movement, they need to be in a body. Okay? They can't survive on their own. So that's the big group for us. Okay? There are some parasites in the other groups, but the big one are going to be the guys who lack movement. Now we talked about, again, specifics with all these fancy names and things for each of the groups. You don't need to know that for this exam. But I can guarantee you all of this is going to come back to haunt you. Okay? So keep it in your brain somewhere. Helminths. What are helminths?
Worms.
Worms, right? So they're large, multicellular eukaryotes. If they're large, why do we consider them microorganisms? Their eggs are tiny, right? So you need a microscope to see the eggs, and that's usually how we diagnose someone as having a helminth infection. And remember, they come in two groups, roundworms and flatworms. Roundworms are round, flatworms are flat. Easy. All right, that's chapter 5.
In the study guide, it says they need to know the life cycle of pinworms.
Nope. Again, the study guide is just all of the information condensed. It's not whittling down anything, okay? So if I skip over it now, 95% chance it won't be in the exam. There's still 5% chance though, because I never will say never. Otherwise, what's the point of having an exam, right? If I tell you everything that's on it. If only I was that generous. All right, viruses. So viruses, again, these are small infectious particles. They are not alive in the strict sense because they do not live on their own. They need to take over another living thing to survive. So this is a nice summary slide of what viruses are. Okay? They can infect every cell that we know of. Okay? So there's millions upon millions of viruses around. And as I said, they are obligate intracellular parasites. Obligate means it's required, intracellular means inside the cell, and parasite means to live off of something that is alive. All right, so the structure of viruses. They're made up of primarily 3 parts: the capsid, the envelope, and the core. What is the capsid?
Protein.
It's a protein coat, and what is it there for?
Protection.
Protection, right, and structure. Next, we have the envelope. What is the envelope?
More protection.
More protection. It covers the outside of the virus. And what is it made up of?
The cell that it was in when it was.
Right, so the cell membrane from the cell that it left from, okay, that it grew in. And then the core, what is the core?
DNA.
DNA or RNA, right? So nucleic acid. It can be DNA or RNA. Will it ever be both?
No.
No, it's one or the other. Good. Out of these 3 structures, which one is optional?
The envelope.
The envelope, right. If you have an envelope, what do we call you?
Oh,
No?
it,
These aren't bacteria, guys. These are viruses.
Is it an envelope virus?
No, other one. They're enveloped, we call them an enveloped virus.
Oh.
Okay? You guys are overthinking it.
I thought you said there was.
If there isn't an envelope, what do we call it? That's a naked virus. Okay, so no envelope is naked. An envelope is an envelope virus. There's no fancy name for it. Alright, so as far as the capsid's concerned, it's made out of protein. These proteins are called capsomeres, and they can be found in 3 different kinds of structure: helical, icosahedral, and complex. What does helical look like? A tube. And what does icosahedral look like? A sphere-y looking thing, right? Kind of like a ball. And what about complex? Complex, right? A bunch of crap stuck onto it, it's all crazy, right? Those are found on bacteriophages. Good. And the main reason for the capsid is structure, protection. The envelope, the viral envelope. Again, found in enveloped viruses. And remember, it's a portion of the membrane, okay, that the virus takes with it as it leaves. And the envelope is there to protect the virus, but also make it easier for the virus to get into a new cell. Okay? Now the nucleic acid. Okay? This is what makes up the core. This is the genetic material. And it can either be a DNA virus or an RNA virus. So again, we talked about a bunch of specifics, right? And again, that was just for your information at the time. We'll talk all about those again later on, but for right now, you do not need to know that for the exam. However, you do need to know the pluses and minuses of being a DNA virus or an RNA virus. So what is the benefit of being a DNA virus?
Oh, well, it can just go in there and infect everything and do the whole job. And RNA is a lot quicker, but DNA takes a lot longer.
So DNA is longer, that's the drawback, right? It takes a while. Because remember, DNA has to go from RNA, or from DNA to RNA, and then RNA to protein, so that takes time. And I think what you were hinting at is that once a DNA virus gets into the cell, it can stay in that cell for long periods of time. So the benefit of being a DNA virus is once you infect a cell, it's there forever, okay? And so that cell can literally be infected for years. So they're long-lived, that's the plus, but they take forever, which is kind of one and the same, but it can be a drawback too. What about RNA viruses? What is the benefit of being an RNA virus? Quick, right? So it goes in, releases its RNA, RNA directly gets translated into protein, and it's done. It's cut out that middle step. But what is the drawback to being an RNA virus? Short-lived infection, right? So for most RNA viruses, they get into a cell, they do some stuff for like a day or so, and then they die, and that's it. But one exception to that are retroviruses. What virus is a retrovirus for humans? HIV. So this is why HIV can stick around for long periods of time in a patient. All right, on to the viral life cycle. As I mentioned before, the viral life cycle is complex. It changes a lot per virus, but there are 6 parts that stay true, okay, for every virus. And that is absorption, penetration, uncoating, synthesis, assembly, and release. So let's start off with absorption. What is absorption?
Specifies on attachment.
Right, so it attaches to the cell, and it does it specifically to the cell it wants to infect, right? So it's got these little proteins on its surface, and those stick to whatever cell it wants to infect at that time. After absorption, we have penetration. What's penetration? Virus enters the cell. Who usually does the entering? The virus, or does the cell bring it in? The cell brings it in, okay, most of the time. Once it gets in, it then goes through uncoating. What's uncoating?
That's when the enzymes and everything, the cell pores on it, and then it removes the capsid, and all the nucleic acids get in there.
Right, so it releases the nucleic acid, and as you said, the cell is what does it. The cell releases crap onto it, trying to kill it. In reality, it just helped it along. Then we have synthesis. What's synthesis? New stuff is made, right? What's being made? Capsid and nucleic acid, right? The 2 major parts of the virus. Then we have assembly. This is where all those bits and pieces assemble together. Remember, this happens autonomously. So if we've got everything together in close proximity, it just snaps together. Then lastly, we have release. Release is where the virus leaves the cell so it can go and infect another cell. If you are an envelope virus, how do you release yourself?
You get pushed out of the cell through.
Pushed out of the cell in a 1-by-1 fashion. This is known as budding. Okay? So you bud out. And you have to do this so that you cover yourself with envelope. Otherwise, you don't get it.
So you would, if you were an enveloped virus where you're being shot out, you're essentially losing that envelope that you had originally? And you
So when a virus comes in, right, the envelope and the capsid get degraded. So all that's left is the nucleic acid.
Okay,
And then the nucleic acid is what acts as the template to make all this other shit.
okay.
So you've got one virus potentially coming in. It makes millions of copies of itself, potentially. And then this forms into the new virus that comes out and covers itself up.
Okay.
So is it acting as like, is it masking itself to get into another cell?
Yep, so the beauty of covering yourself with an envelope is that now you are covered in something that looks like the cell that you want to infect. So when the virus goes to another cell, the envelopes and the membrane look the same and it just merges right in. So that's the main reason they have an envelope. What if you're a naked virus? How do you get out of the cell?
Lysis.
Lysis. So you just cause it to explode. Because you don't need membrane, you don't care, right? So you just explode. This is a nice summary that explains each one of these in, kind of a 1-sentence definition. Again, if you are crunched for time with studying, this would be a good one to look at. What is a cytopathic effect?
Oh, that's when parts of the cell get pretty messed up, like you get 2 nucleases or 2 nuclei.
Right, so the cell gets screwed up because of a virus, right? It's something we can visualize. So some examples of these: inclusion bodies, where you got a bunch of crap in the cell, creates a big hole. Synesthesia, where multiple cells fuse and you get giant multinucleated cells. Or transformation, where the cell just looks funny. It starts doing different things, right? It grows out of control or changes itself. Latent infections is what happens when you get infected, but the virus doesn't actually start to make itself again, okay? So it just hangs out in the cell. The cell acts normal like any other cell does, but it's still infected. What is a virus that infects bacteria called?
Bacteriophage.
Bacteriophage. Right, good. And bacteriophages are a little different than animal viruses. The biggest difference is what?
They inject their nucleic acids in.
They inject their nucleic acid instead of entering the cell, right? So animal viruses fully enter. Bacteriophages just inject. Why? Why is there that difference?
Cell wall.
Bacteria have cell walls, right? So you can't plow through them, they're too hard. So instead you just make a little hole and inject. Good. This is a nice summary between the differences between bacteriophages and animal viruses. So I said the biggest one is going to be penetration, but also viral persistence. So remember, when a bacteriophage injects its DNA, but then it doesn't make virus right away, this is called lysogeny. When an animal virus injects itself or penetrates, and it doesn't make virus right away, this is called latency. So the same concept, but just different names. And remember, for viruses to grow, we need cells. So if we want to do it in the lab, we either need cells that grow in a petri dish, or we need a live animal. Okay, but they cannot grow on their own. And then lastly, anyone remember what a plaque assay is? And why would we use it?
So where it's a monolayer virus that's replicating.
Right, so it's a layer of cells that the virus can infect. You add on your material you think has a virus, and then you wait a couple days and you look for cells that have died. And what those cells that have died told you is that there was a virus that landed there and caused them to die. Why do we do this?
To visualize the presence of a virus.
Yeah, to detect it, right? Viruses are really small, so you can't really tell if they're there. So we have to do this to get an idea if they're present or not. Good. Then lastly, what is a prion?
Infectious protein.
Infectious protein, right? So it's just a protein. There's no nucleic acid. It wiggles its way into a cell and then changes all the proteins in that cell into the prion protein. All right, good. Chapter 7. So, what is a nutrient?
How the organism grow needs a sugar or salt base.
It's anything the organism needs, right, that it has to acquire. And when we talk about nutrients, we usually talk about them as elements, right? So carbon, hydrogen, nitrogen, oxygen, and so forth. Remember, these can be both inorganic or organic. Organic means there has to be a carbon and a hydrogen in the same molecule. Inorganic is anything else. So carbon sources, when we talk about this, we usually talk about the difference between a heterotroph and an autotroph. What does it mean for a heterotroph? How do they get their carbon sources? From organic molecules, from other living things, right? So they steal them from other living things or once living things, right? Even dead stuff still counts. What about autotrophs? Where do they get their organic molecules? They make them, right? They take carbon dioxide and water, which are inorganic, and turn them into organic molecules like glucose. For us, which one has the pathogens? Heterotrophs. Good. Nitrogen comes from the air. However, most organisms cannot get it from the air. Only some can. Oxygen, again, comes from the air. Hydrogen comes from any organic molecule, as well as water. It's very easy to obtain. Phosphorus is a mineral that we get from the soil, or plants get from the soil. And sulfur, again, is a mineral. Okay? So all of these come from easy-to-get places. These are all those trace nutrients we talked about in Chapter 7. And these are the ones that are responsible for acting as metallic ions in Chapter 8, or activating enzymes. Okay? You don't need to know any of them in particular. Anyone remember what a growth factor is? So a growth factor is an organic molecule that the organism cannot produce itself. So it needs to get it from the outside. Okay? Most organic molecules the organism can make. Some of them they can't, and so they have to steal it. We've already talked about autotroph and heterotroph. And remember that we can also talk about these molecules as far as energy sources, okay, or how the organism utilizes them for energy. And by far and large, most organisms are going to be what we call chemotrophs. That means they produce their ATP or utilize the energy from organic molecules. So like glucose, for example, that we talked about in Chapter 8. This is where we're going to find pathogens and by far and large most organisms on here. I've already talked about saprobes, but remember that these are organisms that live off of dead things. And by far and large, most microorganisms are saprobic. Okay? They like dead, decaying material. However, some of them can be parasitic, and that is where they like living things. Okay? So these are organisms that live off of other living beings. And so this is where, again, we would find our pathogen. All right, so those are the nutrients, and now let's talk about how they get into the cell. So why is this a problem, getting things into the cell? What's blocking them from getting in? The membrane, right? So that cell membrane blocks stuff from getting in and getting out, right? So how does the cell get around this? Carrier, right? It makes a hole in the membrane with a protein called a carrier. This then lets the molecule in and out. And what happens when the cell decides it doesn't want to do this anymore? How does it stop it? It gets rid of the carrier. Okay? So it's very simple. Put protein in, make a hole, stuff comes in or out. You want it to stop, get rid of it. Okay? Remove the protein. Diffusion. What is diffusion?
From higher to lower.
Yep, so it's where molecules go from high concentration to low concentration until everyone is equal. Okay? And remember, this occurs in any liquid, okay? And it does not require any energy. Okay? It is a natural phenomenon. When we talk about water and diffusion, we call that osmosis. Why do we give water a special name for its diffusion? What's unique about water, unlike all the other molecules? So it's nonpolar and it can go through the membrane, right, on its own, relatively easily. And so because of that, it can cause problems. So if I have a cell, like this pretend cell here, and I put salty water in it and then stick it into fresh water, what would happen? What's happening as far as diffusion is concerned?
The water spills in through the membrane.
Water rushes in because it's trying to equal itself out on either side. It's not possible, and the cell will explode if it does nothing. Conversely, what if I have a cell full of fresh water and I stick it in salt water? What would happen? Cell shrivels up, right? Water rushes out, trying to equal out on either side. Never happens, cell dies. Okay? So the cell constantly has to worry about osmosis, otherwise it will die. And when we talk about osmosis, we usually talk about the liquid that the cell is in, because that's what determines how osmosis is going to work. Okay? Isotonic means the same amount of salts on either side. Hypotonic means less on the outside than on the inside. And hypertonic means you have more salts on the outside than on the inside of the cell. And again, organisms prevent osmosis from killing them by having rigid cell walls or being able to get rid of the water as quick as possible. Now we talked about how molecules get through the membrane using a carrier. Now let's talk about how that happens specifically. Okay? Let's say I have more molecules on the outside of the cell than on the inside, and I want to bring them into the cell. How would I do that? more molecules on the outside than on the inside. How would I bring those in? Do I have to do anything fancy?
Certain protein carriers have.
So you put a protein carrier in, right, so the molecule can get through it. And then what? And then you do nothing, because it comes in naturally. Why? Because it's going with the concentration gradient. You've got more on the outside than on the inside. If you open up a hole, these guys will just come in naturally until the same amount of molecules are on either side. Why? Because of diffusion. Okay? So if you have more on the outside than on the inside, and you want them to come in, you just put a hole in and let them fall in naturally. What happens if it's the opposite? What happens if you have more on the inside than you do on the outside and you want to bring it in? Can you just let diffusion naturally let them come in? No, because then they'll go out. So what do you do instead? Use ATP, right? You use energy. This is called active transport. So facilitated diffusion is just a hole that lets natural diffusion occur. But it has to go with the concentration gradient. Active transport is when you're going against the concentration gradient and you need to add in energy in the form of ATP to get this going. Animal cells also have another trick up their sleeve, endocytosis and exocytosis. What is endocytosis? Swallowing up stuff. Okay? So wrapping the cell around it. And exocytosis is the opposite, right? Spitting things out. Why can animal cells do this, but plants and fungi cannot?
If they have a cell wall, then.
Yeah, they have a cell wall, it's too rigid, they can't wrap themselves around this. Okay? So this gives them one extra benefit where they can actually bring large molecules into the cell. Alright, so the rest of this chapter is a little dry, okay? A lot of definitions. So, for temperature, this is like first grade stuff. Minimum, maximum, optimal. Minimum is the lowest, maximum is the highest, optimum is the ideal, okay, that an organism can grow in. And for our purposes, the most important group are going to be the green guys, which are the mesophiles. These are the guys that grow at 37, at least that's their ideal condition. Next, gases. And when we talk about gases, we usually talk about oxygen. Because oxygen is required for most aerobic organisms to survive. Now when we talk about oxygen, there's a continuum, right? So we've got guys that really like oxygen. Those are called what? Aerobes. And guys who really don't like oxygen. What are they called? Anaerobes, right? And then we got some guys in the middle, okay? So the first guy in the middle is a facultative anaerobe. What is a facultative anaerobe? So it doesn't necessarily require oxygen, but if oxygen's there, it can use it, okay? So it doesn't need oxygen, but if it comes in, it goes, cool, I'll use this and I'll make energy from it, or ATP from it. On the other side of that, we have a microaerophile. What is a microaerophile?
You just small amount of oxygen.
So low concentration of oxygen, right? So these guys have to live in a liquid where there's less dissolved oxygen. Okay, they can't live with the 20% that we are surrounded by. Okay. And then lastly, the aerotolerant anaerobes. What are these guys? So they don't use oxygen, but it doesn't kill them. Okay? So if it's there, they can survive in the presence of it, but they don't need it. Okay? And they can't utilize it for anything important. And then up here, this is that thioglycate broth that we just used in lab. Okay? So if you're wondering what it will look like next class, this is a good picture for it. All right, ecology. So what is ecology?
How organisms